Good day, welcome back. Today I want to introduce you to some more of the features of the Visual Genome Browser that would allow you to navigate around to specific positions in the genome. The first thing I want to do is to open the controls panel. You do that by going to the controls button and clicking on it, and then it opens a panel slides open that gives you more controls. Today I want to focus on these controls that contains radio buttons. There's 1, 2, 3 and P. Now 1, 2 and 3 allows you to put specific markers at positions in the genome, and P allows you to uh, check for an open reading frame for a protein to see if there is a protein at that specific location. So let us just go back to the full view. So for example, if you want to highlight one position, you just double click, and if you look here on the main genome view as well as on the zoom genes view, you can see there's a blue a circle appearing. You can now go and scroll away from that position. One of two of the buttons that allows you to move away quicker is these up and down arrows to the right. If I click on them, it moves the genome along, going down in the genome. And now I can, for example, click on number 2 here, and I can click anywhere in the display, and it will put a green circle. So in the main genome view, you now see how far the green circle is from the blue circle, as well as in the zoom gene view. If you quickly want to go back to any of these two, you can just select like the blue one and say jump, and it will jump to that specific location. If you want to go to the green one, you select number 2 and you say jump, and it centers the zoom DNA view around the green marker. There's also a third marker, which if you click on it, it creates a red circle. So if you look in the zoom gene view, you can see here there's actually a green circle inside a red circle, and that's so that when they fall on top of each other, they don't obstruct each other. So this is a way to quickly navigate around the genome by just remembering one of these 3 markers. Secondly, if you want to copy a certain part of the DNA, it works in a similar fashion. If you double click anywhere using any of the markers and you move the mouse around, you can actually see how far there's a number just to the left top of the mouse pointer that shows you how far you are away from that specific marker. So if we just go zoom in a little bit again, so you can see here there's this number. If you double click, say for example there, and you're on the marker, it will show 1. And as you move away, you can see exactly how many bases you're away from that position. That allows you to quickly move to a certain number of bases. Let's say we want to highlight exactly maybe 400 bases. And then if you hold down Control and double click, then it actually copies some text for you that's exactly that length. This allows you to easily select text and copy it to this info panel. It also gives you the reverse complement of that, as well as it also calculates the GC content of this specific region in the DNA. This info display actually allows you to highlight certain sequences that are found in the display. So if we copy any part of this and we just hover on top right of every one of these text windows, and you put that text in there, it actually highlights it for you. So that's a way to easily find it. if we clear it again and press escape, then it disappears. So let's say we want to find all the occurrences of GGCC in here. Then we move to that field, and it's going to highlight all the GGCCs. And this gives you a quick way to find text within this text window. And this applies to any text window anywhere. So if we, for example, move to the one here at the top, there is some text, there's a text filter, search field over there. It is in any of the other windows that represent text windows. Okay, so when you've copied this text, let's say we just copy that again. If you copy this text to this info panel, it also copies it to the comparisons window. So if you scroll down here, you can see it's available in the history of the comparisons, and you can get it back by just saying copy there. Another way that one can easily highlight sequences or patterns of sequences of DNA is to use the search box at the top left. And to illustrate that, I'm going to go to the telomere sequence of the genome, which is at the very beginning of the genome. So we move here to the very top, and here you can see there's the telomeres. This is the topmost 5' telomere, and you can see here what the sequence is TAACCC. The telomeres are responsible for protecting the ends of the chromosomes from being rejoined and from the repair machinery incorrectly thinking that it's a broken chromosome and joining it to the end of another chromosome. So it contains a gene in the genome called telomerase, and telomerase will use the telomerase telomere template, which is an RNA gene, and then use that to regenerate the telomeres at the ends of chromosomes. So, for example, if we just select this, we double click there, and we press Control and double click, it copies this telomere sequence to the info display, and then if we put it in the search box at the top, you can see here it's going to highlight all places where that sequence occurs. And then together with that, there's this mismatches at the top here. Let's see if I could make it a little bit bigger. This mismatches allows you to increase the number of mismatches in the highlighting that it will allow. So if I, for example, change it to allow one mismatch, then it's going to cover even more of the DNA. So now if we now move to the other end of the chromosome, we're going to find at the end of the chromosome there's going to be a complementary sequence here. So let's just go to the very end. Okay, so then we double click here, and you can see here everywhere where there's a true telomere, it actually highlights it, but it now highlights it in orange. And the reason for that is because it's actually matching in the reverse complement. So I can also change this back to 0, and then you can see there are less that matches exactly. So another feature of this search box here at the top, there are these buttons just next to it. One is N, F, RC, R, and C. So N basically just means you can select a type of sequence. Now it is currently a nucleotide sequence. You can change it to to being a protein, or to automatically detect. That is just in case it would normally automatically detect what it is. So if I put an M in here, it will automatically detect that it is actually a protein sequence. But if I revert it, it will detect that it's a nucleotide sequence again. F is for formatting it. So if there are any spaces or new lines, you can just format it. And then reverse complement will actually reverse complement what is in there. So if it reverse complements, you can see this changes to red now, because now it exactly matches, and it's not its reverse complement that's matching. And then there's other buttons which allow you to reverse it, or to just directly complement it without reverse complement. So that's some very handy buttons. You can paste any sequence in here and then get its reverse complement using that. Okay, so in addition to just highlighting specific DNA sequences in the DNA, you could also do a pattern search. So if we, for example, want to look for a specific pattern in this highlighted or this display DNA, let us just copy a certain part of it by double clicking and then pressing control double click. So what we could do to search for a specific pattern is you can actually do a wildcard search by replacing some of these with a dot, and that represents any base, A, T, C, or G. So if we now copy this into this search box and we change the number of mismatches to 0, you can see that you can actually do a wildcard search by having any base in that position. Another way of doing it would be, for example, to use brackets. So you could use a bracket to say if a C or a or a T is allowed, or a T is allowed in that position. You can use it in the same way. Then it will still match. If the C is removed, then it's not going to match, as you can see. And if the C is put back, then it matches again. So that means you could do almost like a pattern search with specific bases at specific positions. I believe they sometimes call it the motif as well. Now in order to show you that this box here at the top can be used for searching through the genome as well, there's this find pattern button at the top. What I'm going to do is I'm going to search for an ALU element. Now there is a specific ALU element that occurs many times in a genome, and I've copied it here. It is 288 bases long, so we paste it in there, and then maybe we specify the number of mismatches to be maybe 60 mismatched base positions, and then we just click the chromosome that we want to search in, which is chromosome 1, and then we say find pattern, and it's then going to start searching. It does not use an index in this case. It just simply does a brute force search using the Boyer-Moore algorithm together with the pigeonhole principle to find a certain number of mismatches. So it's still busy, but I just want to point your attention to the fact that I've selected gene regions for the label here, so that does not show gene labels. Otherwise, it's just going to be cluttering the screen too much. It's almost finished. So now it says that it has found 6,062 occurrences in this length of the genome, and then it also gives you the probability of finding so many in this just by random chance. So if you look at a chromosomes view, you can see how many of them were found. So there's truly a lot of repeats in the genome. So if we click on it, you can see it's just littering the genome. So if you right click on any of them, it shows you the specific sequence. It allows you to copy it to the clipboard, each of these as well. So if you double click here, it goes to that specific location. So in this way, you can search for any sequence in the genome and with a specific number of mismatches. The more mismatches you allow, the more it's gonna take time. So there's another way to actually search or try and highlight a specific sequence that you're searching for. When you've entered the sequence here at the top, you can select the alignment view, and then you simply go into the Zoom DNA view and you just highlight where your start base must be, and then it does an alignment, a local alignment against that DNA, so you can see where it mismatches exactly. So let's just zoom that in, so you can see here, if I move the mouse there, it shows you exactly where it matches and mismatches, and then it also displays this alignment in alignment tab. This button's over here, so you can press forwards and it will jump to each of the subsequent search results. Alternatively, you can use the page up and page down buttons. This allows you to easily iterate through all of the search results one by one. If you want to navigate up and down of the zoom panel, you can press the button, this up and down buttons over here. And if you want to make it bigger, then it will jump by the corresponding amount.